Proteins are essential to life and found all throughout the human body, such as the muscles, bones, hair, skin, and more, as they help build, maintain, and rebuild body tissues. Proteins are comprised of amino acids. There are 20 different types that are either made from scratch or by modifying others. The amino acids are the building blocks that help the protein fold into a 3D shape, such as an alpha or beta helix. There are many different types of proteins, some of which are infectious molecules.
Prions are infectious particles that are similar to viruses but are smaller in size and do not contain nucleic acid, meaning that they do not have DNA or RNA. Also, since prions do not contain any nucleic acid, they become resistant to pathogen-destroying procedures as they require the breakdown of nucleic acid. Prions are made up of the cellular prion protein, PRPC, and there are two types, PRP-sen and PRP-res, both of which are made up of amino acids but have different shapes. The PRP-sen prion is made from healthy cells found in the brain. Although its function is not fully known, they are shown to help with communication between neurons as well as sleep and cell death. The PRP-res prion is a disease-causing prion, and when it comes to contact with the PRP-sen prion, it converts it to res. Over time, the PRP-res proteins begin stacking on top of each other due to their abnormal shape, which in turn creates long chains called amyloid fibers. These fibers are toxic and have the ability to kill cells.
Instead of multiplying in the host, prions act as a template and therefore instigate proteins that are folded normally to fold abnormally and take a prion form. These misfolded proteins further act as templates, creating an abundant amount of prions within the tissue of the nervous system. Prions form plaques which cause neurofibrils to become tangled and get in the way of the synapse. Nerve cells as a result get lost and become damaged, creating very small vacuoles in the brain. The tiny vacuoles supply the brain with a sponge-like look when seen under a microscope, giving the name spongiform disease. The spongiform disease, also known as bovine spongiform encephalopathy, also called mad cow disease, causes lesions in a cow's brain, causing them to experience tremors, changes in behavior, aggression, and other various symptoms.
People who consume meat from cows with mad cow disease may develop Creutzfeldt-Jakob disease, CJD. CJD causes prions to fold abnormally. Instead of taking a helix shape, it folds into a flat sheet, meaning the protein cannot function properly. These prions become infectious, an important find as proteins have no genetic material that would allow for reproduction. As more prions misfold and build up, brain cells are killed. As the cells die, the prions enter normal tissue to infect more cells, all without triggering the immune system. Symptoms include motor skill and coordination difficulty and behavioral issues. Mad cow disease caused an economic disturbance in the British beef industry, as many bans to other countries were made and millions of dollars were lost. Since humans pass CJD blood to blood, areas with mad cow disease stop blood donation as well.
Although there has been a lot of research and discoveries done with prions, skeptics still try to prove Dr. Stanley Prions wrong. This is because the idea that proteins can reproduce without nucleic acids goes against everything known about transmissible diseases. The history of prions is quite important. In the 1960s, CJD and Kuru is put into chimps to show the transmissible nature of the disease. In the 1980s, 60 people die after being treated with CJD-infected surgical equipment, and 85 die from getting growth hormone injections that were infected with the prion disease. And in 1982, Stanley Prions coins the term prion, proves that the protein is infectious. And lastly, in 1985, the PRP gene is identified by scientists, and it is seen that those that are not infected can produce the protein normally.
Because of their simplified structures, prions are sometimes called subviral particles. Other types of subviral particles are called viroids, the smaller discovered pathogens. Viroids are infectious RNA particles that are only made up of circular and single-stranded RNA molecules that only affect plants. They are not responsible for encoding protein, but instead replicating continuously when presented in plants. Viroids replicate in the nucleus or chloroplast of plants through a process called RNA-RNA transcription and travel from cell to cell via the plasmodesmata.
Also, unlike prions, viroids only consist of nucleic acid and no protein coat. They use RNA polymerase II to catalyze the creation of new RNA by utilizing the RNA template of viroids, and they reproduce only within the host cell. Furthermore, viroids act as parasites for cellular transcription proteins, meaning that they rely on the RNA polymerase enzyme for replication. These polymerases are able to recognize DNA templates, but are able to only copy virus RNAs.
Viroid-infected plants are the reason for the numerous crop failures that occur yearly, causing a large amount of money to be lost in agricultural revenue due to the diseases that the viroids cause. Examples of important diseases include coconut Prions virus, which causes a critical infection in palm trees, and apple scar skin viroid, which causes the infection that makes apples unappetizing.
Due to the size and structure of both prions and viroids, they are resistant to heat, radiation, and other processes. To destroy them, an agent would have to hydrolyze its peptide bonds to destroy its tertiary structures. However, processes such as incineration, so burning the protein, is a fully effective method in destroying prions and viroids, as it breaks all the peptide bonds and turns them into carbon. Ozone treatment has also been used as a plausible method. However, additional research is still being done to determine its effectiveness.